I'm not telling you how long, but you will see that on this talk. His life was dedicated to tomography. We didn't have a chance to represent proper British stars in tomography, what's going on in our country, but nonetheless, right now you will see through his talk on the whole achievement and what's going on in tomography and how it's successful, and how we can use tomography from the so-called cells to molecules or vice versa, how you can use some sort of molecules to understand what's going on in cells using tomographical methods and describe electron microscopy. Please welcome, we are happy to have you.
Well, thank you, Elena, for the kind introduction. Thank you for inviting me to your conference. So I will speak in the next 45 minutes about different aspects of cryo microscopy. There are indeed different imaging modalities which are shown here side by side on my first slide. The oldest one is electron crystallography, which was developed basically, well, in the mid-70s or so. It's exemplified here by the structure of aquaporin, which was one of the first structures by electron crystallography really pushed to very high resolution. It's a 1.8 angstrom map that was obtained in Fujiyoshi's lab. It really pushed electron crystallography to its limits. Well, as most of you will know, it requires that the molecules to be studied can be persuaded to form two-dimensional crystals. Now, there was a time, I would say, in the 80s when many people felt that one day the world of structural biology might be ruled by electron crystallography. Now, that did not become true, and the reason for that is not that it's not capable to do that, but many of the structures which can be targeted by electron crystallography can also be targeted by X-ray crystallography. And if you have a 3D crystal, have a 2D crystal, I think the winner in the race will always be X-ray crystallography. It's quite a bit faster. The reason for that is basically, well, it's not fully understood, but the yield of images that really go to very high resolution, to atomic resolution, was and is still fairly low. Nevertheless, I think it's fair to say that a couple of important structures, not only aquaporin, if you think of acetylcholine receptor, if you think of tubulin, for instance, have been solved by electron crystallography. Now, I'm not going to talk about electron crystallography today at all, because I have not been active in that field for the past 35 years or so. Then there is what is known or referred to as single particle analysis, arguably a slightly misleading term, because at the end of the day, in single particle analysis, you average over large numbers of individual molecules. Nevertheless, you compare them one by one. And single particle analysis has become a true pillar of structural biology. I think that is fair to say. It's exemplified here by the structure of the ribosome. And the ribosome was, in fact, the workhorse in developing single particle analysis. The obvious advantage is there's no need to crystallize the structure to be studied. And I think the main advantage of single particle analysis is that it can deal with quite a degree of heterogeneity, because you can separate coexisting conformational states or other causes of heterogeneity computationally. It has in the past usually delivered, let's say, medium resolution structures, 10 angstroms or better, which were then kind of upgraded by integrating into it structural information from, many other sources. But increasingly now, with advances in technology, single particle analysis can go directly now to atomic or near atomic structures. And there's electron tomography, and as Elena mentioned, electron tomography has been the main interest of my lab for the past 25 years. It continues to be the main interest of my lab. It's exemplified here by the actin cytoskeleton in this protrusion here. I will come back to that later. I think the advantage of electron tomography over the other methods is that it can study macromolecular or supramolecular structures in situ in unperturbed cellular environments. Now, what I will do in my talk is the larger part, the second part. I will deal with electron tomography. I will show you some technical advances which are really kind of transforming the field currently. But I will also talk a bit about single particle analysis. And I will start with that as a matter of fact. Now, I will talk a little bit about two molecules. Both acting or playing key roles in intracellular protein quality control, protein degradation. The first one is the 26-cell proteasome. Now, there will be a little bit of some redundancy with the talk of Paula Fonseca this morning, but the audience is not exactly the same, and I need to show you a couple of aspects which will be relevant in the last part of my talk. So, very briefly, the 26-cell proteasome is the executive arm, if you like, of the ubiquitin proteasome system, which is the main system for quality control in the cell. It targets molecules that have been modified by the covalent attachment of ubiquitin, usually tetraubiquitin, but well, there are different ubiquitination patterns which have different messages. So I will not talk about that at all. So the machine here basically degrades these substrates, recognizes them, it has to unfold them, it has to deubiquitinate them, it degrades them in an ATP-dependent manner. Generates peptides which are typically something like 8 or 10 or 12 residues long, which are of very little use for the cell, with the notable exception maybe of the immune system, so they have to be cleaved further. And one of the key players in, well, not in all, but in many eukaryotic systems is TPP2, which is a very large complex, quite a bit larger than the 2.5 megadalton 26-cell proteasome, so it's 6 megadalton. But it's nevertheless, in many ways, a simpler structure because it's built from one type of subunit. We'll talk a little bit about that. It generates then, smaller peptides which are finally degraded by an array of aminopeptidases. So let me begin because that was a much simpler approach with TPP2. So TPP2, as I said, is a 6 megadalton complex built from one type of subunit. The building block is a dimer, and so there are stacks of dimers in this complex here. So you can count from 1 to 10 here. So there's a total, each segment here is a dimer. So there's a total of 20 of these dimers which form this kind of spindle-shaped structure here. That was a low resolution map obtained a couple of years ago. We tried very hard to crystallize TPP2. The problem in crystallizing TPP2 is that as you increase protein concentration, as you have to do in crystallization, then these strands begin to grow and you generate very polymorphous structures, which is not conducive to crystallization. So what we have done, our strategy was basically the following. We succeeded in stabilizing a dimer here, solved the structure of the dimer, then by X-ray crystallography, and then we had a map of an electron single particle analysis map of the entire complex here. At the end at a resolution of something like 8 angstroms here. And so given the size, given the quality of the map, given the peculiar shape of the dimer here, we could dock now the structure of the dimer into the complex and obtain pseudo-atomic structure of the entire complex here, which was pretty good quality, I think. Now, I don't want to talk too much about TPP2. I just want to highlight one principle, which is common to all, intracellular proteolytic systems, if you like. It has an N-terminus, which is a subtilisin type. Structure here is located here. That is where the active site is. Then there is a large insertion, which forms a separate domain. Then comes the central domain, which forms the bulk of these strands here. And finally, there's the C-terminal domain, which is, in the dimer, a 20-helix bundle here. And this 20-helix bundle, which determines the exact curvature. And the curvature, determines where the two strands make contact there are some slight conformational changes here which stabilize this particular tertiary quaternary structure here. Now the important thing is that, I mean, in the dimer, these active sites are of course exposed, but they are non-active in that state. So activation follows, I mean, polymerization. And the important thing is that activation, I mean, takes place only, well, as I said, after polymerization, when, I mean, the active sites become sequestered in what we call the catalytic cavity here. And I mean, any substrate to be degraded has passed a couple of constrictions here, shown here schematically. There are two narrow constrictions here, 10 by 10 angstroms roughly. And that makes sure that only unfolded peptides, I mean, can enter, have access to the active site. So, I mean, the fact that activation, I mean, follows sequestration, I mean, protects, I mean, the cell from unwanted degradation or promiscuous degradation, if you like. Now, I will talk briefly about the 26S proteasome. And I mean, as indicated here in this introductory slide, which was meant to become the cover, but the editors decided against it. Anyway, I mean, there are two elements in it. One is, I mean, basically the cryo-EM structure. And I mean, in the approach we have taken, I mean, an equally important element was, I mean, based on cross-linking mass spectrometry, providing sequence-specific information about crosslinks. And so that helped quite a bit in the interpretation of the original map. Now, I will begin to, well, I think there's a need to very briefly introduce the system again. So, I mean, there are two subcomplexes. There's the 20S core complex. That is where proteolysis takes place. And it takes place in a similar way, I mean, to the TPP2 I just showed you. There's a cavity here formed between the beta subunits. That is where the active sites are located. And any substrate to be degraded must somehow, I mean, make its way into that central cavity here. It has to pass a gate here, which is formed by the alpha subunits. And all that, this alone, I mean, is not functional, and in fact, it rarely exists as such, I mean, in a eukaryotic cell, as you will see later on. And so, I mean, it has to team up with these regulatory complexes, which, I mean, first recognize ubiquitinated substrates, remove the ubiquitin, and then unfold and assist in the translocation into the core complex here. Now, I mean, that structure remained, I mean, the textbook structure for quite a while. I mean, as you see, it's, well, not quite 20 years, but a long time ago. I mean, and it was, of course, very unsatisfactory because, I mean, it did not really provide much of an insight into the mechanism of the 26S proteasome. For the core complex, we had a structure, I mean, which we obtained almost 20 years ago by X-ray crystallography, but not for the remainder. I mean, so at that point in time, we decided to leave the field for more than a decade. We didn't see a way forward. Now, what you see here, well, would it still be the challenge to determine the structure of the 20S proteasome today? I think we would certainly do it by cryo-EM. I mean, what you see here on the left-hand side is an EM density map of the 20S. It was just a little exercise. I mean, we never tried to push the envelope here because, I mean, we didn't want really to recapitulate what we had done here before. But just, I mean, this was 3 years of crystallization trials. And this one here was, of course, no crystallization. It was 9 hours of data acquisition, something like 50,000 particles, and then maybe something like 2 days of processing. So, and this is a 4.2 angstrom map. As I said, we didn't push the envelope here. Now, why has it been so much more difficult to work with the 26S? There are two main reasons. One is the sheer complexity. I don't want to bore you with too many details, but maybe very briefly, in addition to 7 different alpha and 7 different beta subunits in the core complex, these are the subunits of the regulatory particle. The prominent subset is the AAA ATPases here. There are 6 different ones. They have non-redundant function. I mean, some have important functions in opening the gate to the alpha, in the alpha rings. Others have more a role in unfolding. Then there are two resident ubiquitin receptors here, RPN 10 and 1013. This little peptide is a kind of a glue, plays some role in the assemblies of part of the complex. Then there are the MPN domain containing subunits, of which one is catalytically active. RPN 11, that is the main resident ubiquitinating activity. Then there are the so-called PCI module subunits here, which have mostly a kind of a structural role. As you will see, they kind of embed these two subunits here and move with them collectively during the functional cycle. And then there are the two largest subunits, which are mostly, pretty flexible adapters for shuttling ubiquitin receptors, but also for ubiquitinating activities. Now, the main problem is, however, not the complexity, but the heterogeneity. And what you see here is a very old map, still very low resolution at the time, but superimposed in this orange color here is a variance map. So it highlights where in the structure the variances are. And you see that the 20S core, and then simplified, of course, crystallization, or also today's structural studies, there are no large-scale conformational changes. Of course, there's some breathing. There has been quite a bit of NMR studies in Louis K's lab, but there's nothing on a large scale that is really resolution limiting. However, very different here in the regulatory particle, where all the variation takes place. Now, we obtained, a couple of years ago, a map at a resolution of 8 angstroms. The data was still collected manually at the time, and so the challenge was to interpret that map and to really, position reliably the various subunits. Now, as I said before, that was kind of augmented and supported by cross-linking. Work done in collaboration with Uwe Ebersold's lab in Zurich, so cross-linking, and then, this is digested. Then, there are ways of enriching, the cross-linked peptides here, subjecting them to mass spec. You end up with the long list of sequence-specific cross-links here. We could use the 20S core to validate the cross-links here, and came up with a kind of a curated list of experimental restraints, as we call them. We did that with different cross-linkers, with different proteases, and we did it with different organisms. We did it for Pompe, for which we had the map at the time, and so we obtained now an interaction map here. We did the same thing for Cerevisiae. We obtained a map which is not exactly identical, which is not surprising, but there's also some redundancy. I should say this one was not used for the modeling exercise, but just for validation purposes later. The cross-linking immediately helped us to establish some important features of the structure, like the order of the AAA ATPases. We had made a prediction at the time, and it was corroborated now experimentally. 1, 2, 6, 3, 4, 5, and that is important because neighboring AAA ATPases form an important structural feature. They form coils together. Now, they also, the cross-linking established in a fairly pedestrian way. We could do that. The relative positioning of these AAA ATPases and the underlying alpha subunits in the outer rings of the core particle. An interesting finding at the time was, which we couldn't explain, didn't understand at the time, that the pseudo 6-fold axis of the ATPases and the pseudo 7-fold axis of the core complex were not aligned. Come back to that later on. Now, not all subunits, provided us with crosslinks, especially those, at the distal ends, like RPN 10 and RPN 13, so the ubiquitin receptors. We determined their position by difference imaging, basically, knocking out RPN 10, RPN 13, or a double knockout. And then, the difference map, you see that in green provided us with a clear indication where the subunits were. Double knockout was perfectly consistent with a single knockout. Now, we used a computational approach for modeling together with André Charlie's lab. So, it's a somewhat busy slide, but the key information is in this row here. So this synthesis, the EM density map was important. I mean, then the absolute localization of, let's say, the receptors, but also some others. Atomic structures of some of the subunits here, homology models of the others, and most important then also the sequence-specific crosslinks we discarded at this stage, as a matter of fact, pre-existing, let's say, proteomics data about interactions, because if you looked at them, there were so many contradictions. Now, the modeling went the following way. We used the EM envelope. We put into it, I mean, beads or spheres, I mean, representing the subunits or domains of subunits here. And then they were kind of jiggled around within the boundaries of the EM map. And for each and every configuration, we scored against the violation of experimental restraints coming from the mass spec experiments. And that provided us with initial information about the localization of the subunits, which was then iteratively refined by molecular dynamics-based flexible fitting here. Now, the outcome of the experiment is described here. We evaluated 1.6 million different configurations for the subunits, quite a number. Of those, only basically 4, I mean, violated either none of the experimental restraints or at most one. So it's almost a unique solution of the problem of the topology of the subunits here. There were exceptions, RPN 8 and RPN 11, they are very close homologs there. We couldn't really say which one was which. But otherwise, I mean, it was, an almost perfect solution. So that led to a model in which, RPN 1, and RPN 2, the larger subunits, one was positioned on the side of the AAA ATPases. That was the position of, as I showed before, of the 2 ubiquitin receptors. The 2 MPN domain subunits, as I said, we didn't know which one was which at the time, were sort of inframed by this horseshoe-type arrangement of the PCI domain subunits. Now, for that, we could put forward already at the time an atomic model, or a near-atomic model, because we had obtained crystal structures of RPN 6. We could do homology models, and since all this is completely alpha-helical, it was relatively easy to interpret. The interesting thing was when we had obtained the crystal structure of RPN 6 here, the crystal packing already indicated that via the PCI module here, they might have this kind of horseshoe configuration. And when we looked at the still relatively low resolution map, 8 angstrom map at the time here, we saw this horseshoe here in the EM map, and so we had already a pretty good guess for that. We could confirm it then more rigorously if you, for instance, take that crystal structure, then low-pass filter it, cross-correlated with the entire structure. You see very sharp and well-defined correlation peaks here, confirming the position in this case of RPN 6 and also for others. Now, in the meantime, technology had changed. Especially, high throughput data acquisition, so we could operate a microscope 24 hours a day, 7 days a week, and we collected an enormously large data set. I should say that the other developments here, like the direct detectors, the faceplate, did not yet play a role in that exercise. Now, that provided not a big step forward in terms of resolution, but nevertheless, a 6 angstrom map is better than an 8 angstrom map, especially if you have basically an alpha-helical structure to interpret. But the global number for resolution is pretty meaningless with a structure like the 26S. So what you see here color-coded, in blue resolution is better, in red resolution is lower. You see, it will not surprise you after what I showed you before, that resolution is pretty high in the 20S core complex, but also in the AAA ATPase part, and it's quite a bit lower in other parts, especially RPN 1, is a convolute of different conformations. So the AAA ATPase, we could interpret in great detail. It summarizes basically the structures for all the subunits here. Most of them are satisfactory. RPN 1, as I just said, is very unsatisfactory because of all these coexisting conformations. You will see later on a map showing, the large conformational changes it undergoes, but we still do not understand how that relates or correlates to the other conformational changes that are going on. And RPN 8 and RPN 11 are particularly unsatisfactory. sheet, and that can of course not be interpreted at all at that resolution. So we focused on obtaining a pretty high resolution crystal structure of the RPN8, RPN11 heterodimer, and we succeeded with the help of an auxiliary nanobody, which strangely enough we had developed in order to have an inhibitor for RPN11, but it bound, as a matter of fact, to the face opposite to the active site, which was very puzzling. And the explanation for that is it becomes inhibitory if you bind it to the entire 26S, it disturbs the structure in a massive way. So it's a less interesting kind of an inhibitor, but it promoted and it helped with the crystallization. Now I should say that the heterodimer as such, the recombinant one, has very low ubiquitinated activity, and that is due to the fact that there is also inhibition by an insertion loop here, which obstructs access to the active site, to the zinc at the active site here. And inhibition is again relieved upon integration into the entire proteasome. So the same story again, that everything is kept inactive until it becomes integrated in a kind of a safe environment. Now, I think I want to finish that part by showing you a little movie that was created by Julio Ortiz in the lab. Basically, just meant to illustrate the structure. It's not meant to say anything about the assembly pathway here. So that is the core complex here, with the 7 different alpha subunits. In come now the AAA ATPase subunits. Some of them, have a C-terminal extension which inserts into a pocket here, and that triggers gate opening here. And you see how neighboring AAA ATPases form these coiled coils, which may act as swinging arms, but their movement is for sure be restricted. Now come in RPN 11 and 8, they are sitting here near the mouth of the AAA ATPase, so RPN 11, as I said, deubiquitinates the substrate. They are enframed by the PCI subunits here. And now I think finally come in, they probably were recruited to the complex late in its evolution. The 2 ubiquitin receptors which sit at the far end of the complex here. Now that is so far still the static picture. And since we had such an enormously large data set of 4 million particles here, we went into some pretty deep classification here. Now what you see here is still morphing, so putting things into a seemingly logical sequence. The challenge is of course related to the biochemistry and to control it. That is well underway, but not yet complete. But all I want to show you here is the magnitude of the conformational changes the complex undergoes during its functional cycle here. So there are large rotations of something, whole modules of subunits by something like 25 degrees, but also shifts. You see it again now looking down the long axis of the complex here. You see that now all the PCI and framing RPN 8 and 11, move, and then there's also a shift. And then we remove, in the next step, we remove, all the subunits except for the AAA ATPases, so to give you a feeling of the conformational changes which go on here, which are related to the translocation of substrate into the core complex here. So I think we have broadly defined the conformational landscape. You should still be aware that what we show at morph here is still, ensemble averages. And I think the challenge is to really dissect it further and to control it and correlate it really with the biochemistry. Very briefly, it's not a lecture about 26F, it's not meant to be. What happens here is that, RPN 11, the substrate and the proximal ubiquitin have to be brought into a position directly above the mouth of the AAA ATPase in what is called by some people the commitment step. Then, degradation becomes irreversible, substrate can no longer escape, and it can be cleaved, which is the prerequisite for translocation here into the core complex. It's a pretty complex Now, I think I want to change gears here completely, but I will return to the topic at the end of my talk. Now, I think I start to introduce tomography a little bit philosophical. I think it's fair to say that traditionally structural biology has taken an extremely reductionist approach. I mean, cells are taken into pieces, the components are isolated, purified, and then they are studied. Now, this has been enormously successful, but I think it's also fair to say that very rarely, I mean, can discrete biological functions be attributed to individual molecules. They are performed by assemblies, by complexes, which come in different flavors, from stable to extremely transient. But I mean, functions really arise from the interactions of the constituent parts of large assemblies, whether they are, well, as I said, stable or exist only transiently. I think it's also fair to say that many supramolecular modules, if you think of centrosomes, if you think of kinetochores, whatever, are deeply embedded in cellular environments, and I think it's more or less impossible to isolate them without violating their structural integrity. I think biochemical pathways are very likely to be organized in a non-random manner, but I mean, these, I think it's impossible to reconstitute them in vitro because, I mean, it's so difficult to mimic cellular environments, including crowding, etc. So I think there's quite an incentive to be able to do structural studies, not in this reductionist approach, but I mean, in an intact cellular context. And that is exactly, I mean, was the motivation for developing cryo-electron tomography, because here the data acquisition is more complex. We have to perform a physical tilt, and in all that we have to keep the electron dose, I mean, very, very low. But we end up with a cellular map, which, I mean, to interpret, of course, can be a challenge. Now, but I mean, the challenge becomes smaller as the quality of the tomograms improves, and you will see there's a steady improvement in the quality. You see here, I mean, this is about 10, 12 years old. This is structure of the filipodium with an actin cytoskeleton from ichthyostelium cells grown directly on an EM grid. So you hardly can recognize the actin filaments here. And this is all because we cannot apply a very high electron dose. We are very limited in that, so signal-to-noise ratio is poor. That means interpretation or segmentation of the tomographs is quite a challenge. We developed the tools to do that, to do it in a fairly automated way. There's usually first a denoising step, and then following the denoising, which is a smart way of doing low-pass filtering, we can generate a template structure, in this case a very short-acting filament, right dimensions, and then, I mean, cross-correlate it, so it indicates where the filaments are located. And then, I mean, the most difficult step is to extract a network by establishing, using a series, I mean, a couple of rules, I mean, which filaments are really connected, and so on and so forth. So, with data of that quality, certainly difficult. Nevertheless, one can do quite a bit, and I will show you only one or two examples. That is from a collaboration with Pascal Cossard's lab. This is about, I mean, Listeria comictans. As many of you will know, Listeria is a bacterium that can hijack the cytoskeleton, the actin cytoskeleton of its host cell. It remodels the actin, forms these cytoplasmic tails or these protrusions, which enable the bacteria to be propelled through the cytoplasm and also to be transmitted from one cell to another. That is what it looks like, and it has mostly been studied by fluorescence microscopy so far. Now, what you see here is a Listeria cell here. Now you see this already better quality. You see as we go through the tomogram, I mean the actin filaments, and this is now the automated segmentation. So we can, from the level of each individual actin filament, I mean, describe its path, and we can describe in a quantitative manner, I mean, the properties of the actin cytoskeleton. I mean, go back one, sorry. For instance, I mean, if you run again, you will see that one property which is very characteristic of these type of actin cytoskeleton is that they form bundles here. Typically 10, 12 or so actin filaments are bundled together. In these bundles they are highly parallel, but the bundles are somewhat misaligned with respect to each other. If we go into such a protrusion to a different field here, this is now the trace of the individual actin filaments here. We can, now for each filament, compare its position with respect to its neighbors. And as you do that, you see that, most of the actin filaments here have a relatively well-defined spacing of 12 nanometers. So if you take into account the thickness of the actin, it means the space between two actin filaments is something like 5 nanometers. There's a little bit of higher order, but not very much in this case, different in other actin systems. The angular distribution, is mostly around 5 degrees or something like that. Now, if you take all the actin filaments which fall into this range here, and superimpose them, you see again, even in this densely packed thing, that it is composed of this relatively highly ordered bundles here. So this is a typical feature of listeria protrusions or comic tails. That is one example. Now, one example, again, that is older work, published, well, that is, our general philosophy, and I come back to that a little bit later on. We can do the same kind of template matching now, of course, also with, globular structures, with complexes, and what we call the thermosome, as we first described it. And you can take now low-resolution structures of that kind, use them as templates, cross-correlate them with the tomographic volume, and then you obtain a set of coordinates describing the position and the orientation of the molecules in the tomogram. Now, that is relatively easy if you apply it to, let's say, something like ribosome. Ribosome is, convenient to work with in the end. It has quite a bit higher contrast than proteins. Now, we went back to a very old experiment done, as you see here, more than half a century ago by Jim Watson and Tissier. They observed that under conditions of starvation in E. coli, in a sedimentation experiment, or centrifugation experiment, they observed there was a new species of ribosomes they called 100S ribosomes. And based on extremely sparse data, they put forward this model that in these dimers, basically they made contact via their small subunits, and that might be linked to translational shutdown. Now the question was, was this observation, in vitro observation, relevant for what is going on in the cell? Jim Watson was not sure about that at all. But we repeated basically the experiment, and yes, under conditions of starvation and doing the ultracentrifugation, we obtained this population, which is composed mostly but not exclusively of dimers here. So the next step is to describe, basically the molecular signature of these dimers here, and that is done here. So if you plot, let's say the center of mass distance, and the angular distribution here, you see that it's, clustered. Of course, not all ribosomes in the population are dimers, but there's a very clear cluster here. Now, okay, you can do subtomogram averaging here, and you can, then dock into it, the known high-resolution structures. There are a few densities which are not accounted for by the high-resolution structure. These are factors which, promote dimerization. Some of them have been characterized in the meantime. But important thing is now the experiment with the intact E. coli cells. So you see tomograms now for cells grown exponentially. You see a tomogram for a cell under conditions of starvation, where you might expect translational shutdown. But you see a tomogram now in which, the cells were first kept in a stationary phase, but then the medium was replenished, so they might, resume protein synthesis. So as you apply now this kind of analysis now to these tomograms here, you see that in exponentially grown cells, there's no indication of dimer formation. If you go to the stationary phase cells, I mean, there's quite an accumulation of ribosomes here. Roughly 50% or so of the ribosomes seem to form dimers here, and minutes after replenishing the medium, the dimers disappear again. So this is, I mean, confirmation that that this dimer formation is something physiological. Now it will become a bit technical. I mean, it's very easy to apply tomography to prokaryotic cells because of their small size. As you go into a eukaryotic cell grown here directly on an EM grid, I mean, then only the peripheral regions of the cell will be transparent enough for the electron beam. But I mean, large parts, actually, most of the volume of the cell will remain inaccessible to tomography. You can do some cryosectioning, and we, I mean, invested quite a bit into it. It's a very tedious and pleasant experiment to do, but that is not a bad thing. The bad thing is, well, first of all, I mean, they do not attach very well. That is a cryosection, a thin one, does not very well attach to the carbon film. But the most serious problem, really compromising everything that is done with cryosections, is a massive compression during physical sectioning. Compression can amount to something like 30–40% of the volume. You don't know whether that, I mean, applies also to the molecules, I mean, embedded to it, which would make subtomogram averaging, for instance, obsolete. So we invested our money into focused ion beam technology, very common in the material sciences and semiconductor field, but it was questionable whether it would work with amorphous ice. So, I mean, we have a dual beam instrument in which, I mean, the scanning electron microscope part is used, I mean, to observe, I mean, the cells, as you see it here, directly grown on the grids. And then, I mean, the focused gallium beam is used to ablate material in order to make it thin enough without any compressions, I mean, for tomography. We were very concerned about a couple of things, not so much about ion interaction or penetration. I mean, it was clear that it would not go very deep, and we used a very shallow beam, so I mean, it would be only a very superficial thing. But we were most concerned about, I mean, temperature rise. But so we built devices to measure it, but we could always show that the dyes remained amorphous or vitreous. Contamination, I mean, is just a technical problem of some kind. A problem which is not 100% resolved at this point in time is specimen navigation, as I call it here. That means if you have a large cellular landscape, to target exactly the structures, I mean, you want to study. Because the size of the kind of windows we cut is of course limited. So the basic experiment is the following. We grow the cells directly on a grid. Then we have our focused ion beam coming in from the side, ablating material from above and below, and leaving behind a thin lamella, which is supported only by the surrounding ice here. We had to take some measures to make that stable so that it didn't break and fall out of the volume, but I mean, all that could be solved, as you will see. You can, in the meantime, and this is just with a couple of volatile cells, I mean, some monas, cut windows or lamella, which measure something like 30 by 20 micrometers here. So we have several cells, as a matter of fact, these small cells here. Then we can select whatever area of the cell we're interested in. For instance, here we zoom in now on the Golgi apparatus. That is what a healthy Golgi apparatus looks like in a cellular environment. This is just the raw data without any denoising, without any segmentation, and it contains an awful lot of information. But the challenge is again to fully use that. Now, we can do it in a targeted way, but we can target things only in X, Y, and not in Z direction. So we have built devices which also have been licensed in the meantime and are commercially available. So we can use fluorescence microscopy operated in a cryo mode and to locate, to identify and to locate features of interest. And then we can go with our focused ion beam to areas we have selected of this cell, and cut them a lamella. I want to illustrate that with something we are currently particularly interested in. That is a project with my colleague Ulrich Hartl. We work on Huntington inclusions, and we want to study the aggregates and the fibrillar networks completely in situ. so in this grid region here, there's a cell which has a nice Huntington inclusion of the right size. We can, overlay that by coordinate transformation, so we know exactly where in the cell the Huntington inclusion is. Then we can cut the lamella here, and then we have in the lamella, in this case, the entire inclusion here. Now, what we can do is we can zoom into any area of this network here, and you see, it's, again, this is the raw data, and it's not yet using all the tricks we have available now, not yet the faceplate, not yet direct detectors, etc. But you see the filtered fibrils fairly nicely. We go now into a region which is, let's say, at the periphery here of such an inclusion. So this is the bottom part where the fibrils are. These are the organelles, in immediate vicinity of these aggregates here. You see that the mitochondria are pretty unhealthy, typical for something which might have a perforated membrane. But that is all remains to be studied in detail. You see an unhealthy Golgi apparatus here. So we can study now these aggregates really in situ in a fully intact environment. Now, still, as I said, interpretation is not trivial because, signal noise and contrast is not so great. So we invest also quite a bit in developing another tool. I should credit for that Agustin Danev. He was, in Kuniaki Nagayama's lab, and he worked for a couple of years in Japan on the, let's say, electron-optical equivalent of the thermal faceplate, which is a very old idea in electron microscopy. Sometimes it's difficult to transform an idea into something that you can really work with. So, he had shown, very nicely at the time that, the faceplate functions as it should function. It produces a cosine type phase contrast transfer function, and contrast is enhanced, but charging and related difficulties were encountered during the faceplate experiments. In other words, you could hardly work with these faceplate. They change their properties all the time. Now, the faceplate as it was used there, the ceramic equivalent was just a carbon film, so that was only in the middle. So that's why enhancing the phase shift and the primary beam, going through the hole, not so easy to align. Many other problems, fringing coming from the sharp edges. But still, nevertheless, contrast transfer function looks quite nice. Now, but as I said, it was even a problem to take a single tilt series without changing the faceplate. So there's a new development, and it will become commercially available very soon. We're still not allowed to talk about all the details, struggling with our industrial partners there. But the bottom line is the following: that we take advantage of something that was disturbing in the past. Namely, that under the electron beam, the carbon film changed its properties. We take advantage of a phenomenon that is responsible for the aging and the non-durable faceplates. We modify it by pre-irradiation such that the phase shift is generated by beam-induced water potential. That is the principle, and it has many advantages. It doesn't need to be aligned and anything like that because it's automatically aligned. It's generated in the microscope, and it avoids many of the problems. So just a few examples of now neurons growing directly on an EM bridge. So we show just the raw tomograms, and you will see that, for instance, here you see the microtubuli, with remarkable clarity and contrast here. We can zoom in, you see, very clearly already the protofilaments here of the microtubuli in the tomogram. You see there are a couple of molecular inhabitants of these microtubuli and so on and so forth. That is another area of a neuron, and if you compare now the quantity, this is mostly actin, cytoskeleton here, so, except for one microtubulus, you see that contrast is now quite a bit higher than it used to be in the past, and segmentation is becoming trivial. There are bundles of actin, and as you zoom in, you see I mean, we see now very clear periodicity along the active filaments in situ. I should say this is not a sample that had been thinned by focused ion beam technology. That is still a complete intact neuron directly on the grid. And as you calculate a power spectrum here, I mean, we have information going to something like 20 angstrom here. And I should say all these experiments so far were done on a TechNight 20. So for the experts, it means it's not an instrument that was in any way optimized for tomography. It was an experimental microscope for developing the faceplate. It was not equipped with an energy filter 200 kilovolts. It had a conventional CCD camera. So, I mean, I think we can go quite a bit further. To give you a little bit of a flavor of things we hope to do. This is at a glance, I mean, this is a little piece of the cytosol of a neuron, and I think it's pretty non-spectacular at first glance. But as you zoom in, for instance, this guy here, now this is a tomogram. I'll slice it to a tomogram of a single 26S proteasome in the context of the intact cell. This is another one here. As you look closely, I mean, you see even, I mean, the internal cavity system in the 20S. So there's absolutely no averaging. This is just the raw data. Now, we can, of course, do some analysis of these data. I don't want to, well, okay, that is now an average of subtomograms of 26S. I mean, we can use, I mean, the methods I described to locate and identify them. I mean, it's so trivial, you see them with the naked eye now easily. I think you will see that, I mean, the two regulatory particles here are pretty non-equivalent in contrast and density. One is quite a bit weaker. So as you calculate for the whole data set of 2,500 subtomograms now, a variance map, you see we have a very strong variance in this region here, which means we have particles which have or have not the second regulatory particle. We have, I mean, well-defined and well-localized variances in the regulatory particle here. We'll come back to that in a minute. So as you take the entire data set and do a first classification, it turns out that that was came as somewhat of a surprise that the typical, almost canonical structure of the double cap 26S is a minority in the intact cell. It's something like 24% of the population. The single capped ones, I mean, are 3/4 of all the particles in the cell. 20S alone is a minority of only maybe 2-3%. Of course, the difference map shows very clearly that is trivial. I mean, there's one missing. Now you can go a step further. You can take the population of single capped ones and you can subject them to further classification. And well, you see in the variance map again, I mean, that is consistent with this one here. That, I mean, the variances in the regulatory particle are very well defined and localized. You can separate them and shown here at the bottom. We cannot separate them yet into the three basic conformations which occur in vitro, but at least to the two dominant ones. I mean, you need a larger data set, one, I mean, corresponding to the commitment state. That is the state when, I mean, the proteasome is engaged with substrate. This is what we call the ground state. These are particles which are sitting there waiting for substrate. Again, I mean, a very well-defined difference map here. You might not easily see the differences here, so we have morphed them here in this one here. Resolution is currently something like 23 angstroms for the in situ studies here, but we can very clearly correlate, I mean, the two conformations we observe with, I mean, those are filtered versions of what we observe in vitro here. So, I mean, what we can do now is we can perform in the cells a kind of a census of capped, 1/4 double capped. We can further say that of the single capped ones, I mean roughly 3/4 again are in the ground state, so they are just sitting there, not waiting for business. Smaller part in the commitment state. If we analyze the data set for the double capped ones, we have roughly, 52% in the waiting state or in the ground state. We have, the other half is mostly, one side is obviously in the commitment state, the other one is not. But it seems that what is going on at the two ends is not directly correlated, so we have also a minority of particles here, which is, in the commitment state at both ends. Now, I think we can refine that analysis quite a bit. I think we have also room for improvement for data quality. But I think the important thing is that we can, for each and every particle, say where it is and then what its status is here. We maintain the information about the localization in the cell here. So as indicated here, that is of course a challenge to take advantage of the fact where are they particularly active and so on. So we can correlate, let's say, informational status with localization. Okay, I think then finally, that is my hope for the future. I think currently something in the 20, 23, 24 angstrom range with intact systems. I think that is if we cannot do massive subtomogram averaging. We must keep in mind that a tomogram typically might contain something like 10 or 12 particles. So it's a lot of work to get a large data set there. But I think, we have not yet combined the advantages really of, let's say, direct detection of phase state and of making thin samples. We can get to quite a bit higher resolution if the sample is, let's say, 100 or 200 nanometers thick, as we can do it easily now with focused ion beam. I'm quite optimistic that we will be able to push the limits of cellular electron tomography to something like a nanometer and hopefully beyond that, so that we can do some real structural homology in situ. So I should say, that is the group that worked on the 26S. It was a very fruitful collaboration with Karen Laska and Andrei Schnali at UCSF. Keiichi Tanaka on the localization of the receptors, and Uli Ebersold on the cross-linking and mass spec. And that is the group of people, that has been involved in the technology developments. The last part, on the in situ 26S, I should mention shortly the collaboration between Shu Asano and Yoshi Fukuda into that. And I thank you for your attention.
Everything from how small particles to the huge cells, and how in general through the huge cells and new technologies and into simple particles.
In a way, yes.
Question, please. Okay, to start your life slightly easy, you have your chance. I would like somehow to ask you a bit more technical questions.
Yeah.
How much is the relevance between the phase space and energy filter? What we would recommend us is energy filter is one of the classes to make that and to see much more thick slices.
I think the energy filter becomes, then important if we have a relatively thick sample. It depends, of course. The energy filter takes out the not so productive contributions from the inelastically scattered electrons. The mean free path for some discussions that are on there, but let's say the mean free path for inelastic scattering in ice is